you go to Canvas and you open the workshop Refractive Index mismatch. And now I'm going to let JJ talk.
Or do I need to show that Canvas web page first, or we can start?
Let me see. I think that they need to open the file, don't they? Let me see. No.
No, later on they need this file, but now they
okay.
can just listen to my
There's no need.
lecture.
No, there's no need. We have all the information here. And I think you saw my email, right? I've split the.
Yes, I saw that.
Good.
I already modified accordingly, so no problem with that.
Good.
Yeah, thank you.
Thank you.
Okay, hello everyone. Here's the refractive index mismatch following service lecture. This workshop will give you more in-depth, hopefully gives you more in-depth understanding and about the consequence of the refractive index mismatch. First, we start with the learning outcomes, and actually this refractive index mismatch workshop can hopefully help you to understand why the image of your sample is never and will never be like your real sample. And we will do the following things during this workshop. First, I will demonstrate you how I acquire images of a sub-resolution fluorescent beads with the single point confocal microscopy, and to monitor the point spread function with the matched refractive index and with mismatched refractive index. We see the shape and size change of it, and then I acquired all the different combinations of this data, and then we will divide you into groups to explore the data using these quizzes. Each group will have 2 questions to answer. One is very straightforward. The other, you need some group work. And then after exploring the data, we will summarize everything and to demonstrate you what happens to the image of your sample if you don't match the refractive index of the sample and the objective. Okay. And feel free to stop me if you have any questions or concerns during my presentation. I will start with the first thing, and I think you're already familiar with this one, point spread function. So I think we already introduced to you that point spread function is the thing that help us to know the resolution of our microscope. And here is the design of this workshop materials that we will use. We will use this red fluorescent spheres, which has a diameter of 100 nanometer with the excitation at 580 nanometer and the maximum emission at 605 nanometer. We got this from Invitrogen. It's a commercial one. And if you want, you can also get the same product for your own microscope to measure your own point spread function with your own objectives. But here we use this fluorophore, and I put this fluorescent spheres into 2 different places. First is on the cover slip, which has the thickness of 170 microns, where this is supposed to be your kind of ideal sample location, and this should give us the best, also give us the best point spread function that we can acquire. And another place is that we have a spacer between the cover slip and the glass slide with a thickness of 50 microns. And I put another layer of the microspheres there. So to mimicking that, if you put your sample there, and what will happen with these samples. And between these 2 layers, we have 3 different conditions. We can have air in between. We can fill in with water or fill in with immersion oil, which all of them has different refractive index. And also for all these 3 different samples, we use 3 different objectives. Air objective, which requires only air in between the objective and cover slip, or water objective or oil objective, to see what's the difference if we imaging with different objectives with different samples. So in combination, we will have 9 different conditions, 3 by 3. And this is to simulate different types of samples with different refractive index imaged with different types of the objectives. This is the experiment design, and this is the equipment that I used. This single point confocal. And the cover slip is between the slide and the objective because it's an inverted microscope. And I use the single point confocal, but you can also use wide field or spinning disk confocal for the point spread function measurement. And first, I give you a overview of this sample. I scanned the 3D volume of this air sample, which means I didn't fill in water or oil in between. And with the air objective, you can see this is the cover slip side, and above 50 microns, we have this glass slide. And I made a small video to show you the in different orientations, how does this sample looks like. Of course, this is only the region of interest. We have even more beads surrounding, but I only imaged this area, which is already more than enough for us for our purpose, because you can see we have so many different beads. Okay, and after getting this sample, and the next question is that which bead should we choose to measure the point spread function? Because one that is outlier from the others is this one, because this one is super bright. Can we choose this one? Actually, if you zoom in, actually it's this one. This one is very bright, and you can see it has much larger point spread function than all the others. And as it is very bright, it will give us very good signal to noise ratio. But in reality, actually, we shouldn't choose this one, because as you can see, it is much larger than all the others, means that it's aggregates. It might contain 2 or 3 single beads. Remember, we only want to use sub-resolution beads. If we have 2 or 3 beads aggregated together, means that this single cluster will have, for example, in total maybe 200 or 300 nanometer in diameter, which is larger than our resolution of the single point confocal, which will not give us the correct results for the point spread function measurement. And also for all this kind of beads, we cannot select because they have very close neighbors, and their point spread function will spread and interact with each other, which also affects our measurement. So what we should choose is all this single and isolated and not surrounded beads. And then after identifying all these single beads, we can, because now we use 28x air objective, now we can switch to 60x objective for real point spread function measurement. And the acquisition, actually, for this, it's kind of straightforward. We do, for the point spread function, because we know it's a dot, we just do a Z stack from the bottom to the top of this bead, and we find the Z plane of this bead, and then move up and down, give enough Z space so that this volumetric acquisition will include all the information that we need for this point spread function. And here is an example. You see that this is Z plane. We are moving from bottom to top. You can see initially we don't see anything because we are far away from the beads. Then as far as we are quite close to the center of the beads, we see the signal, and then it disappears. But we need remember to give enough space below and above so that we can acquire the entire point spread function. Otherwise, you might lose part of it, which gives you incorrect result of the point spread function. And yes, after getting this, and this is acquired that stack with the ontology the view, we can see that the point spread function starts from here and ends around somewhere here. And then we can have the 3D reconstructed view of the acquired point spread function. And this is another movie that help us to see this acquired image. Remember that actually we are imaging a microsphere, which is has a regular shape. But now you can see that we have an elongated shape already, although we are imaging actually on the sample on the cover slip. This is because that the resolution of the microscope in the Z direction is always worse than in the XY direction. As Sylvie already, I think Sylvie already mentioned about this. And so you can see, we can play it again, and you can see that XY resolution is smaller, and Z resolution you get this elongated shape from a microsphere, even with the sphere on the cover slip. And now that actually now what the purpose of this workshop actually is to demonstrate with you the point spread function acquired with matched refractive index and also mismatched refractive index. Here in this condition, we have two different setup. One is the oil objective with the oil sample, means that we are imaging the beads on the slide, which is 50 microns away above the cover slip. And if we have the matched refractive index or with the mismatched refractive index, which means that we have air in between, and we try to see the difference. We are doing the same thing. We are doing the Z scan from far away from the bottom, far away below the microsphere. You can see we can already see something quite early, and then this is matched. We see it disappears, but with this mismatched, we still see something when moving up. And so if we put all this together, we can see that compared to the point spread function on the cover slip, with the matched refractive index, the shape of the point spread function doesn't change much compared to the cover slip. It's maybe a little bit larger, but not that much. But if we compared with the object the air sample, which has a mismatched refractive index, the point spread function is very elongated and with a very irregular shape compared to the one on the cover slip or the one with matched refractive index. This is a consequence of the mismatch the refractive index. And this is just some visual inspection. And what we want to do next is to quantify the difference. And so we already mentioned the point spread function and what we should use to get the quantification result from it, which is, I think, is the relays XY resolution. But here, for this workshop, we will use the full width half maximum, this one, because she mentioned here, it's easier to map for the measurement, and also because we are using this Fiji plugin called Metralo J, which implements this full width half maximum measurement in it. And here is the interface of it. It's quite straightforward to use. We just open the image, that stack image, and then run this plugin from the plugin. And then with the interface, we fill in all the information that is needed, not so much. The microscope type with the confocal wide field or the emission wavelength, and NA of the objective, and also the pinhole used, and click OK. It will generate a PDF report based on this 3D image. And this is a report that it can generate, and it will give the full-width half maximum that is measured based on this image stack. And how it creates this number is that for the X resolution, it will draw a line across the X direction and then fit it with the equation and then get the full-width half maximum of this equation. Similarly, for Y in the Y direction, it will fit in the Y direction, and in Z it will fit in the equation in the Z direction, and extracted the number there. And actually, this is the point spread function quantification results that we get from these different combinations. We have 3 different samples and 3 different objectives, and this is XY resolution and the Z resolution when the sample is on the slide, which means that 50 microns above the curve slip, or if the sample is on the curve slip, which is directly under the curve slip, and also there's a ratio of this resolution ratio. If we divide this one of the slide with the one on the curve slip, we will get this ratio 1 by 1. And you will need this table later on for your small quizzes. And but before going to that, I will give you some more introduction or some more experimental results to demonstrate with you that the mismatched refractive index also leads to wrong Z distance measurement, which Sylvie already mentioned in her lecture. To measure, because we have this sample, actually it's the same sample that we will use, and we already know the real distance between these 2 layer of the beads is 50 micron. But if we use different objective and different media to fill in, we just check what's the measured distance it will be. And to measure that distance with the microscope is also quite straightforward. We just also perform a Z scan or Z stack acquisition. We also scan from bottom to top, acquire from bottom to top, and during this we will see 2 layers of the beads. You see, this is the first layer, this is the second layer, and then after getting this, we will try to find the frame that has the highest intensity for each of the layer, and then get the Z coordinates from the metadata of the images, and then subtract. Then we get the measure the distance. That actually, this is just mimicking how we normally measure our sample with a microscope. Although we already know that the real distance is 50 micron, but if we measure in different conditions with matched refractive index or mismatched refractive index, we see whether there's any difference. Actually, there is a huge difference. You can see this is a summary of this measured distance. If we have the matched refractive index, for example, if you use air objective to measure air sample, or use water objective to measure water sample, or your objective to measure oil sample, we always get a distance very close to 50 micron, the real distance. However, if we use water objective or oil objective to measure the air or water to measure the air sample, we get increased distance. While if we use air objective to measure the water or oil sample, we get very shortened distance. Why is this? Actually, Sylvia already explained quite well in her lecture, but I will just repeat a little bit with a little bit, maybe a little bit of change, or I hope you can understand better, because everything comes with this Snell's law because of this refractive index mismatch. If we use the air objective, for example, with the NA1 and to imaging water sample, which has a refractive index of 1.323. As the n number is smaller than the n sample, which means that the theta here will be not bigger. This angle will be smaller than this angle, but we always assume that the light or also the microscope manufacturer always assumes that we have matched the sample, which means that we always assume that the light goes straight. So we always thinking, oh, although we are imaging the dots here, we always think that, oh, we are imaging something here, which means that this is the measured distance from the cover slip and this is the real distance from the cover slip. So in this way, the measured distance will be smaller than the real distance of the sample. On the contrary, if we have a oil objective which has a higher NA than the water sample, the angle will here of the sample will be larger than the angle here, but we still assume that we are imaging a perfect sample which should have the same angle or the straight line if we don't consider the cover slip. And so this means that the measured distance will be bigger than the real distance. Only if we have the matched refractive index, if we use water objective to imaging the water sample, we have the equal number, and also the angle will be the same here and here. And then the distance, measured distance, will be the same as the real distance. And so only with the matched refractive index, we can get the proper image. And with the higher refractive index of the objective, then the sample, we will get elongated images, while on the contrary, we will get the compressed. Although I think this is not 100% correct, because we will have different, as Sylvia mentioned, we will have a different effect in that as further away from the cover slip. But this is just to demonstrate to you how, in general, it should look. It will be the acquisition. Okay, now this my lecture, my presentation is here, and now it's time for you to do some quiz to practice, and you will need to go to the canvas and try to explore this table a little bit, and I think I will show you, and I think you have 4 groups, and each group will have one quiz, and you just go to the refractive index mismatch workshop and do the quiz that according to your group number.
Yeah, so I've now created 4 rooms, and then I'm going to send you all to different rooms. And so what you need to do is go to Canvas to the after the workshop there, the quiz, objective and refractive index mismatch. Okay.
Yes, I think we might give you around 5 to 10 minutes, because it will be each group will have only 2 questions. First question is really straightforward, so hopefully you can finish within 10 minutes, or just come back to the main room if you finish earlier, or you can explore a little bit if you want for other groups quizzes. They are quite similar, just different conditions.
Okay, so I open all the rooms and see you all guys soon. Bye.
Okay, so I think they're all in.
Sylvie, sorry.
Yeah, yes.
Can Eden and I work in the same room, please?
Okay, you want to be in, so I move you to room 3.
Sorry, thank you.
No problem. And I'm going to move Robin to room 4. Oh.
So Sylvie, actually, I have
Yeah.
a question about your lecture. You mentioned that you always suggest to use the low NA, lower refractive, low NA or low refractive index objective to image the sample with a higher refractive index.
Yes. Yeah, well, it's just no. What I mean is that if you cannot match, you should use an objective that has a lower refractive
Lower,
index than the sample. Because if you use a higher one, then you make everything.
But I mean, because you reason this as that because we will not be able to use the full NA of the objective, right?
Yes, as well. So it's the point also to go higher up if what you're using when you go higher up in the you want to if you go from a water objective to an oil objective because you want a better resolution, but you're actually not going to use the whole NA and everything is going to be elongated.
But if, for example, if you have water sample and you use the oil objective, and
Yes.
because normally the oil objective always has a higher NA.
Yes, but you're not.
Then
You're going to decrease your NA.
you decrease, but I'm thinking whether it will be still higher than the NA of the air objective.
Yeah, so we don't know. It really depends how deep you go in the sample.
Yes.
So,
So
and it's going to be a different refractive index at different places. So
yes,
again, if you're only imaging the surface, it's fine.
yeah. I'm thinking about whether it can still improve a little bit of the NA even if we use the oil objective.
Yeah, I mean, this is really completely dependent on how
Yes.
deep you go in the sample.
Yes, but
But
I mean,
if you go deep in the sample, the NA keeps on reducing.
I mean, for example, for the oil objective, it has have a NA of 1.4, for example, and
If
for the
it's matched. If it's matched.
no, yeah, if it's matched, but for the air objective, it normally is less than 1, right? 0.8
Yeah, so I
is
mean, in general,
it's
the question
quite a large difference. I mean,
absolutely, yeah. But the question is not so much in general if you, I mean, there's a big difference, but your stuff is going to be, again, like, the problem of the refractive index mismatch is going to appear. With an air objective, you have less, it depends if it's a high NA in its class or something. But I think that in general, this is what I've heard, that it's
mm,
better to go lower than higher. And I think
mm,
it is because of the uncertainty that you have different
mm,
conditions at different
mm,
Z levels.
mm,
So if you always image, for example, the same Z level and it's reliable compared to the cover slip, maybe you can compare things. But if you're comparing samples where sometimes you're at that level, sometimes that one, then maybe it's not so comparable.
yeah. Yes, I'm just thinking, for example, only consider about the NA, maybe it's not so bad to using the audio objective. If we consider other things as well, then might not be the case. But for the NA, although it's decreased, it might still higher than the NA of the air objective, for example.
Yeah, so it all depends on the NA of the air objective. If it's a 0.95, it's very high anyway, and without aberrations. And it depends on how deep you go in the sample.
Yes.
So if you're just above the cover slip, there's no problem. So I think it's I think the reason why people say that to go down instead of going up is very much to have something that is like it's a rule of thumb, and clearly you have to adapt this depending on what you're doing, but to have a clear message, let's say. Okay, let's do some room hopping.
Yeah.
Okay, so are you guys finished? Yes, great, great. I think that this goes quick as long as people understand the question. It goes very fast, but sometimes uh, I'll go and have a look. You can have a look in the meantime at the table and try to see what type of patterns you see there that we're going to discuss. Good. So I think that everybody's back now.
Okay, great. Then we can continue with some.
Oh, wait a second. No, not everybody's back. Or?
I think we have
Okay,
this.
yeah, everybody's back.
Okay, now we just continue. I will share my slides and we do some summary and conclusion based on the findings that you just. Sorry, no, sorry. I think I shared the. Maybe I shared the wrong one. I should share the. Yes. So, okay. Okay, I guess now you see my slides and we do some, actually we just use some small quizzes to help you to explore this point scale function quantification results with different conditions. And now we go through together a little bit and then we do a conclusion quite soon. And the first question for all groups actually is quite straightforward. It's about if we have a sample with a mounting medium of prolonged glass antiphase mountain, which has a refractive index of 1.52. And we need to use objective that gives a no refractive index mismatch with the sample. And what kind of objective should we choose? I hope all the groups has the correct answer after Sylvie's lecture and my introduction for the workshop. So answer is the oil objective, as it has the oil objective has a refract with the you need to use oil which has a refractive index of very close to 1.52. And the next question, the second question is different for different groups. We start with group one. Group one's question is that if we have a oil sample that is imaged with the oil objective, which means that we have the kind of matched condition in this combination, and what will be the XY resolution of the sample on the slide or on the cover slip. We start with the cover slip, which is a perfect place for the or the ideal place for your sample. And we just check the table. Actually, we just get this number because we have all your sample with image with all your objective. Just to clarify one thing, this XY resolution is that the average number of the X resolution and Y resolution. Because as I introduced previously, that the for each image, the quantifications plugin will calculate both the the resolution in X direction, Y direction, and Z resolution, Z direction. Here we use XY, the average number of the X and Y direction, just to demonstrate the resolution in the 2D plane. And this is it. And the second question is about the sample if the sample is on the slide, which means 50 microns away from the cover slip, and the resolution or the point spread function of the full width half maximum of the point spread function will be 300 micron. And the difference between this is quite not that far away. The ratio is between of them is 1.04. And similarly for the Z resolution, the resolution in the Z direction for the beads on the cover slip, it will be around 996, and 15 microns above the cover slip, it's 1010, which is also quite close to each other. If this is the condition when we have the oil match the refractive index with oil objective and oil sample. I think I just one team, one group, one submit. I think they all answered correctly. And for group two, the question is about the water sample imaged with the water objective. Actually, the strategy or the extraction method of this is the same. Just we check different locations of the table and find the correct answer. For the XY resolution on the cover slip on the slide, there's no much difference. And similarly for the Z resolution, there's also not that huge difference with the matched refractive index. And for the group three, they have a mismatched sample. A mismatched condition which has the oil sample that is imaged with the water objective. If we have this sample on the cover slip, the XY resolution is 312, while on the slide it is 348. Actually, there's not so huge difference. If we observe the XY resolution, but for the Z resolution on the cover slip, it's quite close to the other combinations. But if you see the Z resolution of the sample on the slide or 50 microns away from the cover slip, it will be 2.4 times worse than the sample on the cover slip. And for the last group 4, they also have the mismatched refractive index condition, which use imaging a water sample with the oil objective. On the cover slip, in the XY resolution is not that a huge difference, but for the Z resolution, it changes from 1,000 micron to 2,000, almost 2,500 micron, which has a 2.48 times difference. So I hope our groups has submitted the correct answer, and just to explore the data. And here I combine all the answer from 4 groups together, and this is a summary of this table. And we can see that with the matched refractive index with either oil objective for oil sample or water sample imaged by water objective, we kind of get not that worse resolution in the Z, X, Y, and Z, even if we have some sample that is 50 microns away from the cover slip. However, if we have mismatched refractive index, for example, if we use water objective to image the oil sample, or use oil objective to image the water sample, we will have very bad Z resolution when the sample is 50 microns away from or far away from the cover slip. Not that far away, actually. It's a little bit far away. The effects starts getting worse and worse as you further and further away from the cover slip. And also, I want to remind you that this is a resolution that I just copied from that table. You'll see how worse it is when the sample is on the slide, which is 50 microns away from the cover slip. If you have the matched refractive index, you are okay. If you are mismatched, you have mismatched refractive index, the results getting worse and worse as the mismatch goes up. But if you have the sample on the cover slip, it doesn't matter much. You have a very good results, even if you're using mismatched refractive index, because you are not your light is not going through the sample or the mounting the medium much.
JJ, I just want to point out something. If you go back here, sorry. Now, if the sample is in water, so the column in the middle, in Z, in the top layer. If the sample is in water and we're imaging with an air objective, we have 2,280 in resolution. And if we image with an oil objective, we have actually a worse resolution with a water.
You mean this one or this
no,
one?
No, so I don't know, you don't see this one. Here, Ruik. Oh, sorry. Here, yes. Okay. So this one, we have the sample in water, and we can compare the resolution of the air objective versus the oil objective. Actually, the oil objective gives a worse resolution.
Yes, that's true.
So yeah, because we had this discussion just before coming here. Oh, maybe anyway, it's a very high. There's a very high difference in the numerical aperture between the oil objective and the air objective. So maybe it's anyway better with the oil objective. But here we can see, I think that you're better off if you have a water sample and you're imaging 50 microns away from the cover slip. You're better off imaging with an air objective than with an oil objective.
Yes, this is a good point. Yes. Okay. And also, I want to remind you that not only the resolution, but the measured Z distance also between the bottom and top beads are also altered if you have a mismatched refractive index that Sylvia already mentioned several times. So here to conclude, in summary, that generally speaking, the Z resolution is worse than the XY resolution. This is something you need to remember, and so the shape might be. So you remember the shape of this point spread function when we acquire the single beads, and also the mismatched objective and sample refractive index leads to worse than that resolution, and also leads to the wrong Z distance measurement if you rely focusing on measure the 3D distance, or you need to be really careful with the refractive index match. So remember to choose the correct objective according to your sample. And if you put your sample on the cover slip, then it doesn't matter much if to which, and then use a high NA objective. Of course, according to your scientific question. Because if you have the thin sample and directly placed on the cover slip, then the refractive index mismatch does not matter. You don't need to consider much. Then you consider other factors for your objective choosing based on your scientific questions. Yeah, I think I'm done. Yeah, if you have any questions, feel free to ask. Otherwise, we are done with this workshop.
So what do you think, guys? What do you think about this?
So we were discussing in the group that now in practice in our, when if you think of our sample, like in my case, for example, I mount my sample with prolonged media, which is oil, but I image with a 10x air objective. So on the XY, we were taking that, yeah, it's kind of the same resolution, but on the set, then the difference is huge. But because in my case, for my analysis, I don't really need the set, then I could get away with it. Or
So
still, it is something to consider.
what is your scientific question?
Is to quantify intensity of GFP signal
And where
in the
do you
cells.
acquire that stack?
Yes.
Okay. So why do you acquire
But
that stack?
yeah, because we want to still capture light, because we work with tissue, which is a bit thick, so we want to acquire different planes. But then when I process the image on Fiji, I do the max intensity projection. And I think at some point during the lecture you were mentioning that that's one way of correcting the
Yeah, I mean,
mismatch, if I'm not.
so basically, so again, it depends on the type of questions that you have, and for what you're doing. So you do your analysis on maximum intensity projection. What I want to point out is that your tissue is on the slide, isn't it?
Yes.
Yeah. So imagine that you're measuring you're doing it the way you're doing with the maximum intensity projection. You have several samples. Sometimes you have more mounting medium than the other. That you have a difference that is introduced because when we go away from the cover slip, I mean, you have seen the low quality of the point spread function when we are mismatched and away from the cover slip. The light is completely scattered. This means the intensity is strongly affected. So if you have one day you have a sample, you image through your sample, you do the maximum intensity projection, and you measure your intensity based on this. The next day, you're going to have a sample with a much thicker, for example, mounting medium. All the intensities are affected. The intensities at the bottom and at the top of your Z stack. So you're introducing some variability in your measurements due to the sample preparation. And does that matter or not? It depends. Guess what? It depends on the scientific question. So if you are measuring, if you're comparing things that are like intensities between samples where you have a huge difference, no problem, right? So
Yes.
when we are measuring big differences, big distances, big anything you want, no problem. You can get away with spitting on your sample if you want. You will get reliable results. But if you're measuring small, you're trying to identify small differences between your mutant and your wild type or something, this is when you're in trouble and you don't really realize it. So
Yes.
that's when it is important to put as little mounting medium as possible or put the sample directly on the cover slip. This is all these things come into play.
Yeah.
So,
And what is
yeah, sorry, I just want to point out because we have a very strong emphasis at the beginning of the course and maybe throughout the course on resolution, the best resolution, It doesn't mean that everybody should get the best resolution possible from their microscope. That's not what I mean. What I want is that you come from your scientific question, you extract from your scientific question the resolution that you need, and you make sure that your machine gives you that, and that your sample preparation gives you that. Your sample design gives you that. This is what we want. It doesn't mean at all that each student should get the best resolution possible in their images.
Right. And what I wanted to ask also related to it, because you were saying like, oh, there would be between days variability on the amount of mounting media that will be. How can you reduce it? Because I always add the same volume of mounting media, but I don't know if, I mean, I guess that's one way that you can reduce this variability, but I don't know if still there's some factors that I cannot control
Yeah,
in
there
terms of
are
how much.
different things, but Gabby will talk about all this on Monday. There are different things. For example, when you do an antibody staining, sometimes you're going to leave more PBS than others another day. So you want to make sure that you remove the PBS from the last wash very well, because otherwise you're going to dilute your mounting medium and you're going to change the refractive index. Maybe it will not dry everywhere. You'll have different refractive indices and stuff like this. But again, all these things, I mean, so Gabby will talk about all this on Monday, and how you can make sure that you have the minimum mounting medium and everything.
Yeah.
But all these things are very important if you are after small differences when you're comparing the
Yes.
different samples, right? This
Okay.
is what I want to stress, that if you have you just need a rough readout, you're okay. So that we
Yes.
know.
Yes. Okay. Perfect.
You're
Thanks.
welcome.
I also had a question, mostly regarding air objectives. Is there any mounting media that would have a refractive index close to air, or if we use air objective, then it's like harder to do matching refractive indexes if we use any mounting media at all?
I don't know how the manufacturers design the optics for air objectives. To tell you the truth, I know that they clearly they consider the cover slip of 170 micron, but I don't know how they work for the rest for inside the sample. I don't think there is any mounting medium that has the refractive index of air, and anyway, your sample would not have the refractive index of air, because we can increase the refractive index. I'm not sure we can, well, sort of. Yeah, if we delipidate a sample, we can lower their refractive index. In general, when we clear a sample, we increase the refractive index to everything. We do not decrease it. So we end up with a refractive index higher than the original sample, not lower. So I don't think you would have any type of treatment that would make your sample have the refractive index of air. Anyway, but also we have to consider that air objectives have a numerical aperture of maximum 0.95 is in practice the maximum. And objectives with a lower numerical aperture are less prone to suffering the image. There's less optical aberrations. So this is one of the beauties also of air objectives, is that we have less optical aberrations with them. So you want to, if they have a very high NA in their class, you can also have some optical aberrations. But in general, they are less prone. So I don't think you have so much problems with an air objective compared to like an oil objective.
Okay, so the benefits of taking care of those questions is lower with air objectives than with any immersion objective.
Yes, except if you have an air objective with an NA 0.95, for example. Like the high NA in their class have also this problem.
Okay, thanks.
Anyone else? Yeah.
I just don't know if I'm not picturing it correctly, but when you refer to putting the sample on the cover slip, so the way I mount my brains is like there's a glass slide, and then I put two cover slips, and then
you put two cover slips.
Like these are just on the side, and then in between those cover slips, I like pipette the brains with the mounting medium, and then I like make sure they're in the right orientation, and then I just put a cover slip on top, like so it's like two and then the one at the top.
So sorry, I'm confused with two cover slips. You have
So
two
like,
cover slips, your sample, and then another
they're
cover
just
slip.
glued to the glass slide, so they don't have any function except to like elevate. Like
Oh,
they
yeah,
provide
and you
that.
use them as a spacer.
Yeah.
Right. So that doesn't matter at all because they do not, the light doesn't go through. You're not imaging through these.
Yeah. But then my question is like, when I put the cover slip on top of the brains, like, how do I make sure, or how do I know that the sample is in contact with the cover slip and there's no space in between?
Yeah. So I think you can come back on Monday with this, but I think there will be a distance because since you have a spacer, you have a fixed volume there.
Yeah.
That you have to fill with mounting medium. So in your design, the way you have designed your sample, it's unlikely that your sample will ever be against the cover slip.
Yeah.
So
Because
we can.
yeah.
Sorry.
Sorry.
I guess this would depend on how thick are her sections.
Yes, and how thick the spacer is. But the sections are never exactly the same everywhere, whereas the spacer is going to be quite reproducible. So I think that we can discuss that again on Monday. How thick are your sections?
They're like whole brains, so
Oh,
they're
yeah. Okay.
200 micrometers.
Yeah. So yeah, we can rediscuss the whole thing there, but I think that if you can put your sample directly on the cover slip instead of putting it on the slide, it is definitely best.
Okay. Thank you.
And you will only see the difference if you compare side by side.
Yeah, that is true.
Yes. Anyone else? Okay, but if we are done, then thank you very much, JJ.